so to continue, again, we're talking about meiosis, or basically the production of egg and sperm cells, which is a reductive division process. So we are not attempting to make identical clones as we were in mitosis. Here, we are actually, basically the goal is to end up with haploid cells that have half the amount of genetic information and are not identical to the original parent cell. We want genetic diversity. So, just a few notes again. So I've been talking about this, but this puts it on paper for you. So in humans, your somatic cells, your body cells, all have 23 pairs of chromosomes. So all your cells have the same genetic information. They're all diploid cells with 46 chromosomes or 23 homologous pairs, right? Where you got one set from each parent. So you got one set of 23 from mom and one set from dad. The only ones that actually can differ would be if you're a male and you have an X chromosome and a Y chromosome. The Y is very different in size from an X chromosome. It's much smaller, has a lot less genetic information. So that's really one of your only outlier in terms of homology. Your other 22 pairs of chromosomes are called autosomes, and again are the same size and have the same genetic composition, meaning that you're going to find the same genes in the same locations, but you have different variants or forms of those genes, which cause all the different traits and different combinations that we have. So again, just to take a look at this picture and the little review worksheet, it's really simple, but it'll have you review this. Homologous chromosomes are matched in length in the position of this. So basically they're going to be about the same size. Your centromere is going to be in about the same position in your two homologous chromosomes. So again, if you have a homologous pair, you got one from mom and one from dad, and as well in gene locations. So loci are your locations for genes. Loci is plural, locus is singular. So again, I'm totally making this up, but let's say this is the gene for having detached earlobes or attached earlobes, right? You're going to find that gene in the same locus on each of your homologous chromosomes, but the actual form of that gene might be different. So let's say this is the chromosome you inherited from mom. Maybe the gene for earlobes you inherit from her are that your earlobes would be attached, and the gene that you inherit from dad is in the same location, but you inherit the gene that those earlobes would be detached, right? So again, the gene is going to be in the same location, but the actual form of that gene might be different, and you'll get a little more into that in chapter 9. And so that's really what I was talking about here, that you have different versions of genes that may be found in the same locus, right? Okay, so meiosis. This is sexual cell division. So we are reducing our chromosome number from diploid to haploid. So we're going from 2N to N. Our goal is to half the amount of chromosomal information that we started with, because again, we need sperm and eggs to have half the amount of genetic info, because we'll eventually combine them. So basically you start life. So when this says begins with a diploid zygote, really what this means is that life begins with a diploid zygote, which is the result of fertilization or the union of a sperm and an egg. Then you would have 2N equals 46, right? But when we start meiosis, the diploid cell that we start with is actually called a germ cell. So we do not start meiosis with a zygote. This is what you get as a result. What we really should say is the end result would be a diploid zygote here. So I don't really like the phrasing of this. It's a little confusing. But again, your diploid zygote is the first fertilized egg, right? Where you combine your sperm and your egg, and that's your first diploid cell that will then be multiplied by mitosis to become you, okay? But to actually go from a diploid cell to haploid cell, in the process of meiosis, we use these special cells called germ cells, which are diploid cells. So here's just a little picture for you to show what I was talking about in the last slide, right? So again, you have basically life starts with the fertilization of an egg cell with a sperm cell. Each of these are haploid gametes, which have half the amount of genetic info, right? So the sperm is N and the egg is N. You combine the two, the sperm fertilizes the egg. That makes 2N. So you had one set of chromosomes in each of these, in the sperm and the egg. When we combine them to make a zygote, now we are a diploid cell. So we've gone from haploid to diploid, right? So we've doubled our number of chromosomes. Now we're at 2N, which would be 46 in humans, right? Then the zygote, or the single fertilized egg, becomes a human by multiplying through mitosis. So basically these cells make identical copies of themselves, right? They multiply through mitosis, okay? So then you have mitosis, creates humans, and then humans produce eggs and sperm, eggs and sperm through meiosis that then reside in their ovaries or their testes and wait for the day where fertilization will occur. So again, when we talk about life cycles, it's just a sequence of stages leading from adults of one generation to the adults of the next generation. So again, with our population, it's really important that we have genetic diversity. So we're going to talk a little bit more about that. But that's why, it's good that we don't reproduce asexually just by making identical clones or copies of ourselves in terms of producing offspring. Our offspring production is sexual, so we do have two different parents and this different combination of genes from two separate entities. And again, life cycles include an alternation between diploid and haploid, which I just went over in the previous slide. So we start with haploid, then we go to diploid, back to haploid, right? As part of the life cycle. So we produce haploid gametes because we don't want the chromosome number to double in subsequent offspring, because as I said, animals cannot survive with any more than diploid sets of chromosomes, right? So if you were triploid or tetraploid, you wouldn't survive as a human. Okay, so let's talk about the stages of meiosis. So meiosis, just as mitosis was, is preceded by interphase. So again, interphase is this preparation stage leading up to division, okay? And that includes G1, S, and G2. We know that G1 is gap 1, or basically a growth phase. The S phase is when DNA is replicated or copied, right, and advanced to prepare for the division that's coming up. And finally, you have G2, which is your second gap phase, which is, again, another final growth phase before we actually start to divide. So with meiosis, again, the special cell that's going to undergo meiosis to become an egg or a sperm is what we call a germ cell, okay? So it is a diploid cell that we start with. So in both meiosis and mitosis, we start with a diploid cell. However, with meiosis, we're going to do 2 rounds of cell division to result in 4 haploid cells, whereas in mitosis, we only did 1 round of division and we ended up with 2 diploid cells. And again, that's because DNA was replicated before the cell divided, right? So DNA was replicated with mitosis before the cell divided, so then we had that identical number of chromosomes in the daughter cells that we originally had in the parent cell, because we had copied the DNA beforehand. In meiosis, we also copy the DNA, but we only copy it once, and then we do 2 rounds of division. So we copy it once, we do a round of division, and then we do another round of division without copying DNA again in order to half the amount of genetic information. So here is just a brief overview of meiosis, right? So you have your pair of homologous chromosomes. In this case, again, N would be 1, and in this diploid cell, this would be 2N equals 2, right? We duplicate DNA during the S phase of interphase, so DNA is copied, and then we undergo 2 rounds of division to end up with 4 haploid cells. And you can tell a major difference here. So in each of these 4 daughter cells, we only have 1 single chromosome. So these are haploid. So N equals 1 for all of these, right? This is a diploid cell. So again, we have a pair of homologous chromosomes here. Here we do not have a pair in any of these daughter cells. We only have a single chromosome. So let's talk about what happens in each of the steps of meiosis. So again, similar to mitosis, we're going to walk through PMAT, so prophase, metaphase, anaphase, and telophase, but meiosis 1 differs greatly from mitosis. When we get to meiosis 2, the second round of division, meiosis 2 really, really strongly resembles mitosis. But in meiosis 1, we have a few things that occur that are different from mitosis Again, because the whole goal of meiosis is much different than mitosis, right? In mitosis, you're trying to make identical copies of your original cells for the purposes of growth, repair, or replacement. In meiosis, where we're making sex cells or egg and sperm cells, we want genetic diversity and we want half the amount of chromosomal information. So we actually want to not produce identical copies. We're trying to produce things that are diverse or different from one another. Okay, so in meiosis 1, the first round of division in meiosis, we're going to have prophase 1 here. So again, prophase, you have some similarities that occur. So your chromatin is going to condense into chromosomes, so your chromosomes will become visible. And your nuclear membrane, as you can see, is starting to break down here, right? And your spindle fibers are beginning to form. Your centrioles are migrating to their poles. So all of those things you're familiar with prophase. Two major differences, though, that occur in prophase 1 are that your homologous chromosomes actually pair up. So you can see here that these homologous chromosomes, so let's say this is chromosome number 1 from dad, and this is chromosome number 1 from mom, right? This is chromosome number 2 from dad, number 2 from mom, number 3 from dad, number 3 from mom. You can see that each homologous pair is actually pairing up. And not only do they pair up, but they actually temporarily are going to cross over each other, right? So they pair up, and where they join is called the synapsis, right? And we would call this temporary pairing a tetrad. And that's because tetra means 4, right? So you have 4 legs here, okay? So again, this homologous pairing is something that differs greatly in prophase 1 of meiosis. This does not happen in mitosis, right? The reason why homologous chromosomes pair up is because it actually enables these chromosomes to exchange some genetic information. So we call this crossing over, okay? So non-sister chromatids exchange genetic material by crossing over. What that means is, again, when I'm looking at this chromosome number 2, I said from dad, right? Remember that this is a duplicated chromosome. So here's one sister chromatid and here's the other sister chromatid, right? So these two we know are identical copies or twins. We're eventually going to pull them apart, but not yet, right? Same here. This is one sister chromatid and this is the other sister chromatid for this chromosome that you inherited from mom. So DNA has been duplicated. So we're not talking about crossing over that's occurring on the same duplicated chromosome. We're talking about genetic information that's crossing over between homologous pairs, right? So some genetic information that was residing on this chromosome here, dad's chromosome number 2, is going to exchange and go onto this chromosome number 2 that was inherited, that will be inherited by mom, right? So we're actually getting exchange of genetic information. So that's why even if you inherit your chromosome number 1, right, you're going to get from the one that you get from dad isn't going to be all dad's information, because we get some of this crossing over or this mixing of genetic information, which again will shuffle some of our genes and just change up the variability in the combinations of genes that you'll inherit. So we call this whole process crossing over. So this again is a super important point to note about prophase 1. So tetrad formation and crossing over occurs. So again, this is just a picture of crossing over. So when you look at what's happening here, right? So synapsis is the pairing of homologous chromosomes, and then the point where they actually join is called a chiasma or a chiasmata if we're talking about plural sites of attachment for that crossing over. So again, you can see here that you have this crossing over that's occurring and the actual exchange of genetic info. So this picture here is going to take you through the same thing. So let's say this chromosome is mom's and this is dad. You get this exchange of genetic information. So again, basically you're getting new combinations of genes or gene shuffling that's going to occur here as a result. Okay, so metaphase 1 in meiosis. Again, in metaphase 1, you're having the lining up of chromosomes in the middle. The one major difference here, though, is that at this point, your tetrads are still formed. So your homologous chromosomes are paired up. So here, a major difference between meiosis and mitosis is that in metaphase 1 and meiosis, our tetrads are lining up along the metaphase plate, right? So here, you can see the tetrads are lined up, and those are what are going to need to be separated.